Far above the seafloor, before there are arms, before there is a star, there is only a drifting shape suspended in open water. It is nearly transparent. It is moved by bands of beating cilia so fine they vanish in the blur of motion. Inside that glass-like body, a gut coils into place. Nerve cells arrange themselves into sensory territories. Muscles begin to contract. And an animal that will one day cling to rock as a five-armed hunter spends its first days as something almost impossible to recognize. Today we're diving into the hidden beginning of starfish life, into the larval stage, where almost every rule suggested by the adult body is broken. The creature we call a starfish does not begin as a tiny star. It begins as a bilateral drifter, with a left and a right side, with an anterior and posterior axis, with a body plan that looks less like its future self than like a message from deep evolutionary time. And that is the first clue that starfish larvae are not merely juvenile versions of an adult. They are a different biological strategy, entirely temporary, ancient, and profoundly strange. When adult sea stars release eggs and sperm into the water, the resulting embryo follows an orderly beginning familiar across many animals' cleavage, blastula, gastrulation. But very quickly, sea star development departs from what intuition would predict. The early feeding larva, called a bipinnaria, is not radially organized at all. It is bilaterally symmetrical, equipped with looping ciliary bands that both propel it and sweep food toward the mouth. It carries a tripartite digestive system aligned along an anterior-posterior axis. In species such as Asterias rubens, the larva develops distinct lobes, an esophagus, stomach, hindgut, and dense ciliary tracts that function like living conveyor belts. It is not a passive speck in the plankton. It is a working machine built for dispersal, feeding, and sensory control. Beneath the apparent simplicity is a surprising degree of anatomy. Researchers have found muscles around the esophagus that drive peristaltic contractions, additional muscles on the aboral side that help shape movement, and an unexpectedly elaborate nervous system concentrated in the ciliary bands, oral region, and apical area. There are serotonergic neurons, oral ganglia, sensory cells, and multiple neurochemical subtypes, evidence that even this transient form is neurologically sophisticated. In other words, the larva is not unfinished. It is exquisitely finished for a different world, the water column, where survival depends on floating, feeding, steering, and sensing, while almost invisible to predators. That is one reason starfish larvae feel so unnatural to us. We keep trying to see a miniature benthic animal, but evolution has built instead a pelagic instrument, a separate body for a separate phase of life. This is where the deeper mystery begins. The larva is not just bizarre in shape, it preserves a historical tension inside the echinoderm lineage itself. Adult sea stars are famous for pentaradial symmetry, yet echinoderm larvae are bilateral. And that bilateral condition is not an accident or a brief glitch on the way to the real body. It reflects ancestry. Echinoderms belong among the deuterostomes, the broader branch that also includes chordates. Their larvae retain a left-right architecture that echoes ancient bilaterian organization, while the adult body becomes one of evolution's most radical redesigns. Developmental studies in sea stars show that this early polarity is laid down remarkably early, even in the egg cell. where components of the Wnt signaling pathway become asymmetrically localized before the embryo divides. Later, that hidden polarity helps establish the axis of the larval body. So the starfish larva is doing something extraordinary. It is living out one body plan while preparing another. The bilateral larva is not simply discarded evolutionary baggage. It is a successful solution to life in the plankton, and likely the ancestral feeding condition for echinoderms. The ciliary bands, the larval gut, the neural patterning, the transparent drifting form, these are not improvisations. They are old, deeply conserved features. In fact, the bipinnaria form appears so constrained and so effective that researchers comparing widely separated sea star lineages have argued it has remained remarkably conserved over roughly 200 million years. That conservation tells its own story. For all the visual strangeness, this larva works. It works so well that natural selection has kept returning to it, even while adult sea stars diversified into predators, scavengers, coral eaters, and reef dwellers. The larva is the oceanic phase where geography is crossed, gene flow happens, and the species expands beyond the reach of crawling adults. It is an ancient vessel for movement through a world too large for the sea star body that comes later. Then comes the most unsettling transformation of all. In many sea stars, the bipinnaria gives rise to a later larval stage called the brachiolaria. This new form is still bilateral, still planktonic for a time, but now it begins to carry the tools of settlement, an attachment complex with sticky larval arms, and an adhesive disc. The larva can test the seafloor before it commits. This alone is strange enough, a drifting creature equipped with temporary glue and provisional limbs meant only to end its floating life. But internally, the real upheaval has already started. The future sea star does not simply enlarge from the whole larval body. Instead, major adult structures emerge from internal rudiments associated with the coelomic system, especially on the left side. In sea stars and other echinoderms, the pentameral body plan begins as a localized developmental project, not a whole body scaling up. In the direct developing sea star Parvulastra exigua, researchers tracked this process in detail. By about 6 days after fertilization, a 5-lobed hydrocoel, the core geometry of the future pentaradial body, has formed. Soon the juvenile rudiment becomes visible. Tube feet appear. The oral side of the developing juvenile turns toward the substrate. And as metamorphosis proceeds, the larval body is not neatly shed like a costume. It is gradually resorbed. Larval tissues are consumed, reorganized, or abandoned, while juvenile structures take over attachment and function. That is why starfish larvae feel almost grotesquely improbable. The animal does not grow into itself in any straightforward sense. It builds a second architecture inside the first. One body feeds and swims, while another assembles on one side, waiting for the right surface, the right cues, the right moment to take command. When settlement comes, the sea star is not merely maturing. It is undergoing a controlled biological replacement. And even that is not the end of the abnormality. Some sea star larvae do things that sound less like normal development than survival tactics improvised at the edge of possibility. In the northern Pacific, planktotrophic larvae of the ochre sea star, Pisaster ocraceus, were shown to clone under favorable feeding conditions. In the crown of thorns sea star, one of the most consequential coral predators on Earth. Larval cloning has become more than a developmental curiosity. Researchers found that when food is abundant, a single larva can split, producing two larvae that then regenerate the missing parts. In laboratory observations, the process was so fast it seemed almost instantaneous, mouth and body separating, each half rebuilding what it lacked. This matters because crown of thorns outbreaks can devastate coral reefs, and larval cloning offers a way to amplify larval supply during phytoplankton-rich conditions. Another study solved a different mystery for years. Strange cloning sea star larvae drifting in the Caribbean were known, but not what species they belonged to. DNA barcoding finally traced them to an Indo-West Pacific sea star, suggesting that larval cloning may help certain species endure long dispersal routes or ecological barriers. Then there is regeneration. In Pentaradial miniata, larval sea stars can regenerate major anterior structures, including components of the nervous system, after severe injury. Experiments showed that after decapitation, larvae can restore serotonergic neurons and re-establish neural patterning by reusing developmental programs and even respecifying cells into new progenitor states. This is a profound clue about echinoderm biology. The larval body is not only specialized for feeding and locomotion, it is also unusually plastic, able to repair, rebuild, and in some cases reproduce asexually while still in the plankton. For a creature we usually imagine as a slow adult on the seabed, the larval stage is almost alarmingly dynamic, capable of multiplication, of reconstruction, of opportunistic responses to feast and injury. Once that hidden life is seen, the diversity of sea star larvae becomes even more revealing. Not all sea stars keep the ancestral feeding bipinnaria for long, and some lineages have deleted it almost entirely. Among astronid sea stars, evolutionary shifts toward non-feeding development have happened repeatedly. Some species develop through yolk-rich, abbreviated larvae. Some settle through highly modified brachiolaria-like forms. Some brood their young internally, reducing larval structures until the boundary between embryo, larva, and juvenile begins to blur. What this means is that the larval stage is not a fixed prelude. It is an evolvable arena where natural selection can shorten, exaggerate, simplify, or repurpose whole developmental programs. Yet even in this diversity, the logic remains biological, not theatrical. Every larval design answers a question posed by environment and life history. Disperse widely or stay close. Feed in the plankton or rely on yolk. Risk the open water or protect development in the parent. Sea star larvae therefore reveal evolution acting on one of the deepest levels available to it. Not just on adult anatomy, but on the timing, sequence, and architecture of an entire life cycle. They are evidence that a species can be shaped not as one body, but as multiple bodies arranged in time, each adapted to a different ecological problem. The adult sea star that grips rock with tube feet and opens shellfish with slow force is only one phase of that design. Before it becomes that benthic animal, it has already been a transparent bilateral feeder, a sensory drifter, a glued explorer of surfaces, and in some species a self-duplicating fragment in the plankton. So when we say that nothing about starfish larvae is normal, what we really mean is that they expose how narrow our idea of normal has been. We tend to trust the adult form, to treat it as the animal's true identity. But sea stars do not grant us that comfort. Their larvae remind us that an organism can be bilateral and then radial, planktonic and then benthic, temporary and yet intensely specialized. It can carry an ancient ancestral geometry in one stage and invent a new one in the next. It can feed with ciliary bands, settle with adhesive arms, grow a 5-rayed body from one side, clone when food is plentiful, and regenerate when broken. And even now, much remains unresolved. We still do not fully understand how some larvae decide to clone, how ecological signals are translated into these developmental choices, how far larval plasticity can go in the wild, or how exactly the genetic choreography of metamorphosis produces one of the strangest body plan transitions in the animal kingdom. The sea star larva drifts at the edge of what our categories can easily hold, not juvenile, not adult, not merely transitional, but a living argument that evolution works across whole life histories, not just finished forms. In the open sea, almost invisible, it carries one of biology's quietest revolutions, a reminder that the creatures we think we know may begin in bodies we would never guess, and that much of life's real invention happens far from the forms that finally settle into view.